Hi everyone, we are going to talk about oxidative phosphorylation. The amount of ATP that human beings require to go about their lives is 2000 kilojoule for a day's worth of activity. To provide this much energy requires 83 kilograms of ATP. However, human beings possess only about 250 grams of ATP at any given moment. The disparity between the amount of ATP that we have and the amount that we require is compensated by recycling ADP back to ATP. Each ATP molecule is recycled approximately 300 times per day. This recycling takes place primarily through oxidative phosphorylation. So you can look at the figures shows up here. Mitochondria, a state grain, form a network inside a fibroblast cell. Mitochondria oxidize carbon fuels to form cellular energy in the form of ATP. Oxidation and ATP synthesis are coupled by transmembrane proton fluxes. Electrons flow from NADH and FADH2 through 4 protein complexes to reduce oxygen to water. 3 of the complexes pump protons from the mitochondrial matrix to the intramembrane space. The protons return to the matrix by flowing through another protein complex, ATP synthesis, powering the synthesis of ATP. Thus, the oxidation of fuels of the phosphorylation of ADP are coupled by a proton gradient across the inner mitochondrial membrane. Collectively, the generation of high transfer potential electrons by the citric acid cycle, their flow through the respiratory chain, and the accompanying synthesis of ATP is called respiration or cellular respiration. Oxidative phosphorylation takes place in mitochondria. Mitochondria are oval-shaped organelles, typically about 2 mm in length and 0.5 milliliter in diameter, about the size of a bacterium. There are 2 compartments in mitochondria. So first, the intermembrane space between the outer and the inner membrane. And second, the matrix, which is bounded by the inner membrane. Mitochondria are simultaneous organelles that live in an endosymbiotic relation with the host cell. These organelles contain their own DNA, which encodes a variety of different proteins and RNAs. Mitochondrial DNA is usually portrayed as being circular, but recent research suggests that the mitochondrial DNA of many organisms may be linear. The genomes of mitochondria range broadly in size across species. The mitochondrial genome of the Proetist plantum consists of fewer than 6,000 base pairs, whereas those of some land plants comprise more than 200,000 base pairs. So let's talk about the respiratory chain. Well, it consists of 4 complexes. Electrons are transferred from NADH to oxygen through a chain of 3 large protein complexes called NADHQ oxidoreductase. So Q cytochrome C oxidoreductases and cytochrome C oxidase. A fourth large protein complex, called succinate Q reductase, contains the succinate dehydrogenases that generates FADH2 in the citric acid circle. Electrons from this FADH2 enter the electron transport chain at Q cytochrome C oxidoreductase. The above complexes are also called complex 1, 2, 3, and 4 respectively. Two special electron carriers fuel the electrons from one complex to the next. The first is Q enzyme Q, so also known as ubiquitin because it is a ubiquitous quitin in biological systems. Ubiquitin is a hydrophobic quitin that diffuses rapidly within the inner mitochondrial membrane. Electrons are carried from NADHQ oxidoreductase to Q cytochrome C oxidoreductase, the third complex of the chain, by the reduced form of Q. Electrons from the FADH2 generated by the citric acid cycle are transferred first to ubiquitin and then to the Q cytochrome C oxidoreductase complex. The reduction of ubiquitin to ubiquitol proceeds through a semiquinone intermediate. Iron sulfur clusters in iron sulfur proteins, also we called the non-heme iron proteins, it plays a critical role in a wide range of reduction reactions in biological systems. There are several types of iron sulfur clusters unknown. In the simplest kind, a single iron ion is tetrahedrally coordinated to the sulfurous groups and the 4 cysteine residues of the protein. A second kind contains 2 iron ions, 2 inorganic sulfides, and usually 4 cysteine residues. A third type contains 4 iron ions and 4 inorganic sulfides and 4 cysteine residues. The electrons of NADH enter the chain at NADHQ oxidoreductase. The initial step is the binding of NADH and the transfer of its 2 high potential electrons to the FMN positive group, yielding the reduced form called FMNH2. The electron acceptor of FMN, the isoalkoxide ring, is identical with that of FAD. Electrons are then transferred from FMNH2 to a series of iron sulfur clusters, the second type of prosthetic group in NADHQ oxidoreductases. Electrons flow in complex 1 from NADH through FMN and a series of iron sulfur clusters to ubiquitin, forming Q2. The charges on Q2 are electrostatically transmitted to hydrophilic amino acid residues that power the movement of HL and BH components. This movement changes the conformation of the transmembrane helix and results in the transport of 4 protons out of the mitochondrial matrix. Electrons flow from ubiquitin to cytochrome C through Q cytochrome C oxidoreductase. So, that's all for this section, and we will continue to talk about it. So, thank you for your attention.